This is just to talk about how we can use statistics and data visualization to identify errors and explore errors that we may see in our study data. And I think the examples are simpler than perhaps some of the other talks. I think some of the other talks have talked about some advanced machine learning methods. And so if you've never gone down this path to look at data in this way, that may be a bit intimidating, but I think any sort of methodologies you can put in place to start reviewing your data is certainly better than nothing. The idea here is that we'll try to use statistics to uncover some unusual data, and because this often requires interacting with a number of people outside of statistics, so people in clinical, regulatory, different departments who tend to fear statistics. Using data visualization certainly makes this an appealing way to communicate things, particularly because there's often a large amount of data we're trying to explore. So our goals are trying to identify these problematic data in our clinical trial. But beyond identifying it, we also want to spend some time trying to understand why this unusual data may have occurred. And so identifying is the first part of it, and sort of the follow-up thing is the investigation of understanding how these things could have happened. And this often takes a little bit more work and may take some effort or some research for things that are outside of the study database. And typically in identifying issues with data quality, we may exceed some statistically relevant threshold if we're using statistical tests to identify problematic data. We may try to predefine some meaningful thresholds that would be important to see even in the absence of statistical significance. And statistical significance often can be potentially a barrier for understanding signals because we may have sites that have few numbers of patients. So understanding how patients are distributed between our sites and how large sample sizes may be at individual sites are certainly important. But also identifying these issues may just boil down to when results do not align with our expectations. So if we do an analysis and it conflicts with what we've seen in the literature or just doesn't make sense based on our scientific understanding of the problem, there may be some investigation that's required into what's going on. And of course, when we look to explain or understand these differences, some of this may be due to natural differences among patients or techniques that are applied within individual sites, and there's certainly methods to try to adjust for that or account for site-to-site variability and how certain techniques are applied. But there may just be carelessness in processes or sloppiness on the account of how people are entering information, or there may be natural ways that manufacturing defects, for example, that can cause certain machines to not function or certain procedures to perform suboptimally. And of course, misconduct is one that I think has typically been in a lot of the literature in terms of people trying to cheat within a site or not filling out things correctly intentionally. So this is a straightforward example of just summarizing some data. So this is from a hospital system where they're trying to understand the number of medication errors and how they can improve quality in terms of the medication errors that they see within this particular hospital. So the first thing they've done is essentially count up the number of different kinds of medication errors. And this is a Pareto plot that's essentially just an ordered bar chart compiling the different reasons of why things have gone wrong. The black line is essentially the cumulative proportion of these events across all error across all of the errors that are taking place in the database. And the Pareto plot's a simple sort of non-statistical way of trying to solve the vital few errors versus the trivial many. So the idea of this 80-20 rule, if we can solve most of the issues with the fewest amount of steps or the least amount of effort, this will greatly improve our quality in terms of how we collect our data and report on our data. So in this particular example, patients missing doses, overdosing, taking it at the wrong time or the wrong drug would certainly be places to vastly reduce the number of errors that have occurred within this particular hospital. So one item that has, or one statistical technique that's used quite frequently in trying to understand differences between patient populations and how different one patient is from another is Mahalanobis Distance. And this is similar to our examples that when we go back to elementary school and we're trying to compute the distance between 2 points in a plane. Here, we're trying to compute the distance between 2 points or 2 patients in n-dimensional space. And rather than assume that these points are independent, we can use the covariance matrix of these variables to help weight the importance of the contributions of the distance to the distance between any 2 individuals. So essentially, we're taking into account the correlation among the variables for particular patients when we're computing the distance. So we can compute distance between individual patients, or we can take a different approach where if we look at the multivariate mean or the centroid of where we would expect the typical patient to show up in n-dimensional space, we can calculate that distance, a patient, how different that patient is or how far away they are from that centroid of the data, so the center of the data cloud. So we can have 2 considerations for this. So obviously, if we're thinking about outliers and we're talking about how extreme a patient may be, I think most of us are pretty familiar with the concept of outliers and how far a particular point is away from the mean of the population. And in the multivariate setting, that works in a pretty similar fashion. But we can also, in this particular example, be concerned about patients who lie too close to the center as somehow being unremarkable. So the argument being that if someone's going to make up a particular patient, they could be considered too good to be true and would be unusually close to the centroid of the data. So it's pretty straightforward to calculate Mahalanobis distance from the centroid, so we can identify these inliers and outliers and develop thresholds of severity to triage the signals that we can identify. And we can do this without a lot of statistics or numbers. We can generate a plot and create a threshold to show where the individual points are looking quite a bit different. So this is an example in phase 2. I believe this is the one of the CDISC sample trials in 254 patients with moderate Alzheimer's disease in 17 clinical sites. And among these patients, there were 44 covariates measured at baseline, so age, vital signs, and a whole host of laboratory measurements and items from questionnaires. So the goal is to kind of get an overall assessment of whether or not patients may be viewed as extreme from one site to another and considering the totality of their data. So we can generate a plot like so, where each column essentially is representing, along the x-axis is representing a different site. And the distance that we see computed along the y-axis, this is the Mahalanobis distance that we've summarized in the analysis. And you can see that generally within the 99 and 95th percentile confidence intervals that most of the patients are falling within these regions that seem pretty straightforward. But within this plot, I've highlighted a couple of points. These are the two individuals that, these are the two largest outliers that are showing up within this particular data set. So we've identified them as outliers. So the question then becomes, well, why are these particular points outliers? So a question we were often asked for these multivariate outliers, are like, this is fantastic, but it's not clear to me why these people are unusual or how I can better understand why these particular patients are unusual. So it being in a multivariate setting, now we have to kind of go back to the univariate setting and understand which covariates are contributing large values to the squared distance. And we can do this and compute these components, but the components themselves don't really have a particularly useful or meaningful scale, but we can compute them. We can compute the proportions of basically saying this is the proportion of the outlier or the distance attributable to a particular covariate. So we can do some transformations of the computations to get these PIJ. And what we can do with these PIJ, we can essentially create a Pareto plot similar to what we did in the first example when we were talking about hospital medication orders. Except here, what I'm doing is I'm ordering the x-axis according to the contributions of the individual contributions of the covariates to the particular outlier distance for a subject. So this is the most extreme subject, this Pareto plot. And what it's showing is that the glucose values and the basophils and the ALT values are contributing quite a bit to this patient being considered unusual. And for most of the other covariates, we're not really seeing much of a difference. And if we were to plot some plots to better understand what's going on in the different spaces, we could see in an individual plot for glucose that, or glucose crossed by basophils, we can see in a more graphical or a more common graphical setting that we have in fact identified some of the features that make this individual unusual in the context of the rest of the population. So this is sort of a straightforward way to start with a multivariate outlier and then identify for each individual subject which features may require additional review by a clinical team. We don't necessarily have to look at individual plots per se, but they could follow up on those particular data points that are seen as problematic. And we could summarize the information in such a way that once we split up once we identify the different patients that are, say, contributing or having large variables that are contributing to their residuals in a very meaningful way, we can create this heat map, for example, to see if there's certain covariates sort of across the board that are contributing to patients being an outlier. To maybe answer the question, is there a particular measurement that's really throwing things off, and that measurement might be attributable to a particular procedure or piece of equipment that may have been faulty. One of the professors I interacted with at UNC, she was involved with a study where the thermometers that were sent to certain region were miscalibrated, and the patients were all, their temperatures were reading essentially a couple degrees cooler than regions in the rest of the world. So this could potentially help identify those kinds of things. Funnel plots are also a pretty straightforward way of identifying outliers, and also considering the sample size of the site that we're talking about. And typically, when we talk about thresholds in some of the older transcelerate guidelines, we talked about thresholds that were really based on sort of a clinically meaningful difference for a particular site compared to other sites that may be within the trial. So this example here, we're talking about deaths. If this individual's death rate at a particular site is within 5% of the overall rate, we may say this is a mild issue. If it's between 5 and 10%, this may be a moderate issue. And if it's excessive, greater than 10% of the overall rate, we would flag this as a severe issue. And the nice thing about funnel plots, typically we may try to just calculate the proportion of deaths in a bar chart and look at them by site. And that's what I've done here on the left. The overall death rate is the reference line that's indicated on the y-axis. So we may be tempted to just say, well, for these first couple bars, or bars that are, say, above 50 or 60%, we may want to investigate all of those. But here's where the statistic comes in, in trying to do this a little smarter. We wanted to look to see, well, what would be unexpected based on the sample size that occurs at a particular site. And the classic example of this is if you have a site with a single patient in it, you essentially have two options, a 0 or a 1. So you either have everyone dying at 100% or no one dying at 0%. So if your goal is to review sites and you spend your time on these sites, that's not necessarily meaningful. So we want to spend our time on the signals that are considered unlikely given the sample size that's available at a site. So if we look to the funnel plot on the right, the x-axis is essentially the sample size of patients at any given site, and the confidence bands are the 95 and 99.8% limits that indicate the distance or the confidence interval from the overall mean. So if a particular site on the y-axis where we plot their proportions of death fall outside of these, then we can consider them extreme based on the sample size at a particular site. So site 6 at the top has roughly 10 patients, but they have 90%, roughly 85 to 90% of their patients dying in this particular example. And that's why those 3 outliers are highlighted in the bars to the left, whereas some of the other ones are still in orange because they're not falling beyond the extreme limit. So we can use these funnel plots within a schizophrenia trial for some examples when we talk about discontinuation rate. Now, just to illustrate, if we use some of the traditional transcelerate methods, we're really just talking about a consistent rule, regardless of the sample size that may be observed within a particular site. And this is an easy way to do it if we don't have statisticians available, or we have a lot of sites that have a small number of patients. But this is essentially what we're doing when we apply this threshold without considering the sample size. So in this particular example, the thresholds without considering sample size is to the left, but if we use the forest plot or the, excuse me, the funnel plot, we can reduce the number of signals in this particular analysis. And when we consider the sample size and the confidence intervals, the funnel plot on the right, we haven't identified any severe signals, whereas on the left, we have identified a number of severe signals. And I just want to make a comment about relative and absolute scale. So I think we tend to, within our trials, think on an absolute scale. So we talk about looking at errors within a particular time frame of a study. So we may want to look at our quality within the first 25% of patients or the first 50 or 75% of patients, or we may be talking about looking at quality from month to month. And this is sort of an absolute interpretation of the quality. And what happens here is that, sites come on to the trial at different times, and we may be experiencing sites that have been on the study for quite some time and sites that may have just started that month. But I think this is typically how quality is assessed, but we could also consider a relative interpretation of assessing quality and sort of scale things to when the site first came online and compute within site study days to see how is the quality for the 5th month of site A compared to the 5th month of all the other sites that have participated in the study. So these are just different ways of interpreting the data and perhaps assessing the performance of quality in the analysis. So these are I showed one particular funnel plot, assuming perhaps that we were looking at data within a single quarter or over the course of a trial. We may want to do this for each time window. So I have this cell plot here that along the x-axis is a number of sites and time windows are along the y-axis. We may want to look at how things change over the course of time, for example. So the one plot, the coloring in the cell plot is essentially classifying patients according to or sites according to where they would have fallen in the confidence intervals of the funnel plot, whether they were moderate or severe or mild events or mild risk, essentially. And we can look at that change over time. So here, for example, we have a funnel plot where we're looking at time in an absolute setting in site 26. If we're comparing to this particular set of time according to the other ones, it looks like site 26 has had issues with its quality in the first 2 settings in the first 2 months of the trial. But if we look at a relative interpretation, if we talk about their first 60 days, it looks like they had some initial trouble, but then that trouble has not continued into the next 60 days based on lack of available data. Essentially, there's no discontinuations. So their quality in terms of the number of discontinuations has improved. I want to talk real quickly about some other analyses that we can do really quickly. So digit preference, looking at the trailing digits of the data that we collect from site to site. So if we're talking about vital signs or laboratories, oftentimes people may round digits and or compute values in such a way that there's some underlying bias. So we can treat each site as a suspect site and compare it to the reference of all other sites and just look at this distribution of this trailing digit and compare it to the reference and compute a statistic. And so naturally this creates a large number of plots. So if we're computing this for each site, for each particular finding in the study, that creates a large number of tests represented by the dots in here. And you can summarize this easily with a volcano plot. So the main idea of the volcano plot is that we're summarizing statistical significance on the x-axis, or the y-axis, excuse me, and clinical significance on the x-axis. And points that have large statistical and clinical significance will go to the upper left or right corner. So you can see here I've highlighted 2 sites based on their systolic and diastolic blood pressure. And if we look at these 2 plots, we can see that these sites are reporting zeros in the digits quite a bit more frequently than the other sites. This may be due to the site perhaps strictly rounding down more often than should be, or they may be using a regular blood pressure cuff as opposed to an automated cuff. But you would need to determine whether or not this is important enough within your study, if this impacts the results enough to go chasing this down and perhaps correcting it. You can see here that essentially they round to either the 0 or the 5 for the last digits of the blood pressures. You can also look at pairwise relationships with correlation, and this could also apply to serial correlation if we're looking at measurements across time within patients. So you can calculate this test statistic comparing the relationships of 2 different variables within a site and summarize these test statistics in a plot as well to identify any unusual values. And you can see here that although I've highlighted 1 point sort of up at the top, but there's a large number, 5 or 6 different pairs of variables that are showing up as being significant for site 28. I can also summarize this and highlight those paired cells here in this analysis. And you can see on the right-hand side that the correlation between these variables, these pairs of variables, is quite a bit more extreme than they are for the overall reference for this particular site. And if you're to plot it, you can see that there are a number of outliers for some of these values that have been created or calculated when we've taken the lab results for people in this analysis. So this is helping drive this pairwise correlation due to these outliers. And it turns out a number of these outliers are due to a flare in the patient's liver disease at the time when this was calculated. We can also compare visit schedules in a similar way. Keeping patients on track and making sure that they attend their visits on time is certainly important. And we can do this in a way that's certainly similar to our computations of digit preference to see if the patients are basically staying on track at one site relative to the other sites in performing the analysis. You may also here identify sites that essentially have scheduling that looks to be too good to be true, where they are essentially showing up perfectly according to the schedule laid out within the protocol. So this was quick and rushed, but I wanted to show a number of different examples where we use data visualization to summarize a large number of statistical methods to identify data quality issues. And the goal of this is to try to use these data visualizations to help communicate what's going in the trial. We certainly can't look at the results of a large number of tests, so we need to rely on these visualizations to help point us to where the issues may be residing and potentially using other data visualization methods to help us understand why these things may be happening in the first place. But in working with anyone who's not a statistician or statistical programmer or a fan of statistics, any way that you can sort of hide statistics and make people sort of rely on them without telling them they're doing statistics is certainly an easier thing to make happen than to tell them we're going to use statistics in our day-to-day work. So a lot of these methods can also be used for safety as well. But I'll conclude here, and if there's any questions, I'm happy to take them.